Welcome back, Bio30 Prep. Working on Unit 1, Properties of Matter. We're still looking at the properties and classification of compounds, and in this lecture we'll be looking specifically at acids and bases. By the end of this lecture, you'll be able to identify the chemical formula for common acids and bases. You'll be able to identify the chemical and physical properties of acids and bases. You'll be able to define the term indicator. You'll be able to describe acids and bases in terms of their pH, and you'll be able to describe neutralization reactions. One of the reasons we're going to be looking at acids and bases in this course is because in biology, acids and bases are extremely important to human biochemistry. They're involved in our digestive process. They act as digestive agents. They help us break down nutrients. They also act as chemical switches. Catalyst is the fancy term, but there's often the pH, or the amount of acid in different parts of our body, determines which chemicals get to do their job inside of our body. So extremely important in digestion. They're also involved in the oxygen and carbon dioxide transport in the circulatory system, and they even help control our breathing rate. So it turns out that the acid level in our blood actually influences when we inhale. So acids and bases are extremely important in biology. In terms of properties, acids and bases do exhibit different properties than strictly ionic or molecular compounds. They're often identified by using pH and indicators, and an indicator is just a substance that undergoes a known reaction in the presence of a particular substance. So these are usually chemicals that, for acids and bases, change color in the presence of an acid or base. Litmus paper is an example of an indicator. It determines if there's an acid or base. If you take a look at our table here, here it shows you the reaction with litmus paper. Acids turn blue litmus paper red, whereas bases turn red litmus blue. If you do stick red litmus into an acid, it will stay red. If you stick blue litmus into a base, it will stay blue. Other indicators can be used to more accurately determine pH, and pH is really a measure of acidity. So, litmus paper will tell you if something's an acid or a base, but it won't tell you how acidic or how basic a substance is. Other properties of acids and bases, acids taste sour. If you want to think of, where's my pen here? Things like lemons, limes. They contain citric acid and they have a sour taste to them. Bases taste bitter. In terms of touch, acids tend to be tacky, whereas bases are slippery. I've already mentioned their reaction with litmus. Another key indicator of acids is they tend to react with metals. So, for example, if you stick a magnesium strip into an acid, you'll end up producing hydrogen bubbles. There's no common reaction with metals for bases. In terms of conductivity, both are highly conductive. So those are the general properties of acids and bases, makes them a little bit different than either the molecular or strictly ionic compounds. Many acids and bases are commonly used substances, things like vinegar, lemon juice. These are acids, whereas baking soda and bleach are bases. Most of the cleaning products we use in homes are either acidic or basic. Acids form solutions with water with a pH below 7. So when we're measuring pH below 7, those are acids. Bases, when you dissolve them in water, have a pH above 7. pH is also a measure of the acidity or alkalinity. Alkalinity is another word for basicity or basicness. So alkalinity of a solution. The lower the pH, the more acidic the substance. The higher the pH, the more alkaline the substance. So something with a pH of 2 is much more acidic than something with a pH of, say, 6. For indicators, often there are chemicals that are indicators. So here's just 3 examples. Bromoethyl blue, phenolphthalein, and methyl orange. All 3 are chemicals that have actually been designed to change color in solutions with a different pH. So for example, for bromoethyl blue, if you put it in a solution with a pH of less than 6, you end up with a yellow solution. pH greater than 7.6, you end up with a blue solution. If it's in between the two, you get a mixture that is green in color. For phenolphthalein, the pH range is a little bit different. If it's a pH less than 8.2, so anything that's acidic or even a little bit basic, phenolphthalein will not turn color, it'll stay clear. If it's between 8.2 and 10, it'll be a light pink color. And if the pH is over 10, so if it's a strong base, I shouldn't say a strong base, but a concentrated base, the color will be a deep pink. So bromoethyl blue blue kind of tests kind of the same as something acidic or basic. That's what its color change will indicate. Whereas phenolphthalein, it's more of how basic is a solution. Is it only slightly basic, or is it really basic? On the other end, there's a solution called methyl orange, and it changes color at very low pH, so it detects very concentrated acids from not so concentrated acids, and it goes from kind of a red to a yellow with this kind of orangey color in the middle. So those are just examples of indicators. You don't have to memorize any of them. You just have to know that different indicators can be used to test different pH levels. Other forms of indicators, pH meters. It's a digital meter that actually gives you the pH of a solution when you stick the sensing rod into that solution. There's pH paper that has a whole bunch of different colors depending on the pH. So you'd stick a strand of paper in. It starts off usually as being a green piece of paper in this case that detects neutral substances. And the more blue it gets, the more basic. The more green or red it gets. Once you get down to red, you get pHs that are quite low. You can have pHs of 0. You can have negative pHs. You can even have pHs that are high, 15 or 16 or something like that. So most pHs are between 1 to 14, but pH can be negative, or it can be greater than 14. And again, here's just another example of what another pH paper that has 4 different colors that you match up, and when you find the right set of 4, you get the pH that you're looking for. For common substances, here's just some examples of what kind of substance has what kind of pH. Battery acid, for example, very acidic. Its pH is around 1. Stomach acid is a pH of 2. Once we get into things that are more common, acid rain, the stuff that melts cars or melts the paint off of cars anyways, 5.5. Here's a bunch of common substances, milk, blood, saliva, all getting close to that neutral range. Some stuff is a little bit acidic, some stuff a little bit basic. And then we get up on the other end where we have seawater is actually a little bit alkaline and has a pH of 8. Hand soap, pH of 9 or 10. And then ammonia or bleach, you start getting to the pHs that are getting close to 14. So down here you'd have very alkaline. There we go. Up here, these are very acidic. And then once you get past about a pH of 3 or so, I mean, obviously, lemon juice and vinegar, they're safe to touch and their pHs are down around 2. One thing we should note is with a pH scale, every every time, and I'll put the word unit here, every unit change of pH that we make represents a 10 times increase in acidity. And here's what that means. If we take a look at, say, acid rain and milk, they differ in acidity by 1 point. Acid rain is 5.5, milk is 6.5. What this means is acid rain is 10 times more acidic than milk. And if we take a look at something like blood, blood is almost at 7.5, roughly. If we'll round it to 7.5 just for argument's sake, that means that acid rain is actually 100 times more acidic than blood. Because it's 2 units, so 10 times for one step, 10 times for the next step, so 100 times. More acidic than blood. So a 6-point step would be 1,000,000 times, 12-point step would be 1,000,000 times. So there's a huge difference in acidity when you compare, say, battery acid and even human saliva. Battery acid is almost 1,000,000 times more acidic than human saliva or blood or substances like that. When we look at acids, it turns out that acids have 2 distinctive chemical formulas. The first formula are called hydrogen acids or H acids. When their formulas are written, there's an H written at the front, followed by some kind of anion or negative ion. So some examples include hydrochloric acid, sulfuric acid. This is battery acid. Hydrochloric acid is actually stomach acid. Nitric acid is another acid. All of these are H acids. They have the hydrogen in front, followed by some kind of anion, some kind of negative ion. The other type of acids are ones that end with COOH. These are carboxylic acids or organic acids, and examples of these ones are things like vinegar, citric acid, lactic acid. All of these things end with a COOH. So those are the other types of acids. If you see a COOH at the end of a chemical formula or an H at the beginning, you're dealing with an acid. For bases, they have a distinctive formula as well. They end with OH, and this is where people get kind of confused. So you got to look at the full formula. Something like NaOH, caustic soda, it ends in OH. If you had something like CH3COOH, this here is an acid. Sorry about that. This is an acid because it ends in COOH. So there's a difference between ending with an OH, just an oxygen and a hydrogen, a hydroxide group, and a COOH, which is a carboxyl group. So you got to look at the whole formula just to make sure you don't make that mistake. Some other examples: ammonium hydroxide, magnesium hydroxide. Ammonium hydroxide is often used as a cleaning solution. But those are some common bases. When acids and bases are put in water, this is what makes, I guess, acids act as acids and bases act as bases. When you just look at water, the occasional water molecule dissociates into hydrogen ions, and this is the stuff that makes things acidic, and hydroxide ions. This is the stuff that makes substances basic. So a very small number of these water molecules break apart into their constituent ions. Now, because there's an equal number of each, water is neutral. So when the number of hydrogen and hydroxide ions are equal, the solution is said to be neutral. And this is an example would be So pure water, even though it makes both of these ions, since there's an equal amount of each, it's a neutral substance. What acids do is they release extra hydrogen ions into solution. So when this hydrochloric acid enters water, it breaks apart into its two ions, hydrogen ions and chloride ions. If we look at vinegar, vinegar is a CH3COOH, it does the same thing. It releases these hydrogen ions into water. And this is because of hydrogen bonding at work. So the role of acids is to increase the hydrogen ion concentration. Bases do the opposite. They increase the hydroxide ion concentration. So the more hydrogen you have in a solution, hydrogen ions, the more acidic. The more hydroxide ions you have in a solution, the more basic. pH is actually a measure of the concentration of hydrogen ions. So low pH means that there's a high concentration of hydrogen ions, and a high pH means there's a low concentration of hydrogen ions, and that's what pH actually measures. Neutralization reactions, bases and acids, because they make these either hydroxide or hydrogen ions, if you get a lot of them together, they start getting back together to form water. And as that happens, a solution becomes neutralized. You're using up the hydrogen ions, you're using up the hydroxide ions, you end up with water molecules, and water molecules are neutral. So, from this lecture, you should get the idea that acids and bases have unique empirical properties that separate them from being either considered strictly ionic or strictly molecular. Acids and bases are extremely important in biology. They regulate processes or the progress of reactions in living organisms. Acids in solution have low pH. Bases in solution have high pH. So pH is a measure of hydrogen ion concentration. The other way you can word it is if the pH is high, you have a high concentration of hydroxide. Acids and bases neutralize each other to produce water molecules and an ionic salt. And indicators are used to determine the presence of an acid or a base in a solution. So from this lecture, it's relatively short. You don't have to know a lot about acids and bases, but mostly what you're looking for is recognition. That when you see a formula, you can tell it's acidic or basic, and that's really about it. So as long as you can see what an acid or base is, and the idea that pH measures how acidic or basic a solution is. That's it.